Alright, so one of the tools that you're going to be using a lot in this semester is a program called Blast. And so the intent of this particular recording is just to get you into the beginnings of how to use this tool. It's a major tool involved in genomics and molecular genetics. It works by finding similarity between sequences, whether they be DNA sequences or protein sequences, and relies heavily on the GenBank database as well as other databases to get information out of. It's used in gene discovery, gene identification. That's where you're going to be using it, used to identify conserved and therefore likely important regulatory regions. You may be doing that in some of your independent studies toward the end of the semester, and a variety of other things. So what I want to do today is just give you a sense of what is BLAST, where do you find the software, what gets blasted in terms of what you're going to be working with, and how do you use the output. So BLAST is a major way into the GenBank sequence database, and that's a large database maintained by the National Institutes of Health in just outside of Washington. And just to give you a frame of reference, that there's something like over 225 billion bases in the database at this point as the sequence database, and then there's also more recently a whole genome sequence database, and that has like another 800 billion bases of sequence in it. So it's a large amount of information that BLAST allows you to tap into and find out things about the genes of the phage that you're going to be working with. You aren't going to necessarily need all of it, but it's nice to know it's all there. So for this course, there are basically three major entry points into getting BLAST. One is at phagesdb.org, and I'll show you a little bit about that. One is through DNA Master, and I will tell you a little bit about that, plus you'll have that from your annotation guide. And the final one is the mothership of blasting, the NCBI, or National Center for Biotechnology Information Blast Server, which is at NIH, and where all the sequences by and large are stored. And I'll tell you some exceptions of that. So switch over to some live information. So one of the things that one of the places you can find BLAST is at phagesdb, at the actinobacteriophage database. And there are two ways into this. One of them is you get to the home page, you see right up here at the top it says BLAST. And there are two types of BLAST: BLAST-N for blasting nucleotide sequence, and BLAST-P for blasting protein sequences. You have another way of getting into it, and some of you may have already discovered this as well, which is that when you go to the page for the phage that you're working with, you will come across something that looks like this, right? And so this is the page for Sally Special. And you probably went down, you looked at some of the things here, you downloaded using the link to the FASTA file to get the sequence down. But there are these four buttons up at the top, three of which we'll cover in a different talk, and the other one is this locally blast this genome. So BLAST, and I didn't say at the beginning, but it stands for Basic Local Alignment Search Tool. And so what it's going to try to do is take the sequence that you give it and align it to whatever database you're pointing it towards. For the BLAST server at phagesdb, it points us to the phage database that's maintained there. So in other words, all the phage that have been isolated and sequenced at University of Pittsburgh, that includes the phages that have come from programs such as the one we have at Maine. So when you get there, you click on it, you can see it automatically fills in the sequence, and it gives you several buttons to push. It has some things down below that later on, when you learn more, we can use to control the BLAST search a little bit. Mostly I'm using that to point out that as you get to these different websites, it's always worth scrolling down to see what else is there. The program we're going to be using is BLAST-N because this is a nucleotide sequence. It's just got A's, C's, G's, and T's. There are some other aspects that we could use. We could translate that sequence and use it to explore other databases, but we're just going to do this one. and all you have to do is click on the BLAST icon or button, and you come up with something that looks like this. And there's a graphic display, and if you look at this bar up here, when I put the mouse over the top line, you'll see that it's Sally Special. So what that is, that's the alignment of the Sally Special sequence to itself. And then the next most closely related phage is Emperor, and the two of them are part of the same cluster. Other than that, there are other phage, but there's only small fragments of them and much lower levels of sequence identity that the BLAST server is giving you. If you scroll down, you can see the listing of them, starting with Sally Special at the top and other ones. And you'll get information out of this in two forms. You get the score that has been obtained by doing the alignment and the statistical likelihood that it has, what's the probability that it's a legitimate alignment. And the lower the number, the better the likelihood is. In other words, the lower number means there's a very low probability that it's not, the two sequences are not related to each other. If we were to look and see what we got out of this, so query stands for your sequence that you put in, and subject is the one that it lines up to. Of course, in this case, they're the same. And the output you see with the alignment is the basis for one, the basis for the other, and every time there's a vertical line, the bases are the same. And that's no surprise. And we have an alignment of all 15,896 bases at 100% identity, the two to each other. So that's no surprise. It becomes more interesting when we start looking at what else is there. So if we instead look at Emperor, which is the next most closely related to Sally Special, then we see, first of all, a couple of things. We see only 86% identity. We see that there's some gaps in this sequence. There's 5 of them. And the score, of course, is much lower. The expected value, the statistics on it is still pretty strong. If we look at the alignment, first of all, we see we're missing 7 bases here. We don't know why that. So, the people working with Sally Special should go back and look at those 6 bases. And we also see places where the bases are not lining up, where we have a C in one and an A in the other, or a G in one and a C in the other. And we also have places such as this, where there's a single hyphen, or here, where there are 4 hyphens. And those places are called, that process is called hyphenation, and that is to keep the overall sequence in a line. If we didn't do these 4 dashes in here, or if the computer didn't do it, then we would have these 2 G's lining up, but this T would then line up to that C, that G would line up to that A, that G would line up to this C, and yeah, these 2 C's would line up, but the whole sequences would be out of register, and it would look like they were completely unrelated to each other, because computers just simply take what you give them and say, well, they're not related anymore. By putting in the hyphens, we now keep the registers the same, so that we continue to see alignment along the way. This process is called hyphenation, and this contributes to the score. So you get a positive score every time you have bases matching up, but every time you have to put in a hyphen, you lose some points. And if you have to expand this gap that you've created, then you lose even more points. The other thing I want to point out here, and this is going to be important as you look at these pieces of information, is remember Sally Special is 15,800 and some odd bases, and Emperor is 16,604 bases. And what this is telling us is the alignment is only 1,600 out of 1,800 bases. So what happened to the rest? Well, what happened to the rest is that when sequences start becoming less and less related, then BLAST splits them into multiple sequences. So here's the first, 1,800 bases, 1,855 bases. But then here's another segment, a segment of about almost 800 bases, where there is alignment between the 2 phage. And again, we see places where there are empty spots. But all it's saying is that when you look at the output, you need to look at more than just what's right there at the very top of the page. You need to continue to scroll down to make sure you have all the information covered. So that's the first entry place into how we use BLAST. And we use BLAST because it's as a way to find out what else is our phage related to. In this particular case, Sally Special really only has one other phage that's related. Cuke has no other phage that it's, because it's a singleton. Well, it has some relationship, but it has very little relationship to anything else. TBRI 12 and Shere-Khan, both are parts of the E cluster, so they have multiple phage they're related to, and we would see that in a BLAST alignment. So BLAST you're going to find 3 different places: phages, DB, DNA master, and then the major one at NCBI. Here's the link for the NCBI one. It gives you many more things to look at NCBI, but the one thing it doesn't have is many of the sequences for the phage that lie in repository at phages DB. So BLAST is an alignment program. It allows you to align your sequences to any of these other things in the database. It has multiple subprograms. The major ones you're going to use are BLAST-N and BLAST-P. Phages DB is a BLAST server just for the Actinobacter phages. DNA master blasts all the protein coding regions that have been identified in your phage genome, and NCBI has a much greater capacity to do alignments, but it's limited by what's in the database. And with that, I will stop.